Alright, so now moving on to lecture part 3, slide number 51. So now in the microbiome setting, for example, stool for gut, you extract community DNA, not culturing cells, fragment it, attach adapters, and yield a library for sequencing. Extraction method matters. It must lyse diverse cell walls fairly. For example, gram-positive bacteria, or your observed community will be skewed. Bias born here propagates downstream. Illumina overview and 4 canonical steps. Illumina sequencing proceeds as sample prep, fragment DNA, ligate adapters with indices, and flow cell binding sites. Then secondly, cluster generation on a flow cell via bridge amplification. Third is sequencing by synthesis with fluorescent nucleotides. And fourth, primary analysis to call bases and qualities. The design choice, many copies per fragment, is why Illumina is both high throughput and accurate. Now sample preparation where multiplexing and compatibility are decided. So adapters contribute index barcodes. So you can pool many samples and primer flow cell compatibility. So clusters can form and be read. This is also where you make size selection choices that shape insert length and thus downstream assembly and profiling performance. Cluster generation and sequencing by synthesis, signal engineering. Bridge amplification turns each immobilized molecule into a cluster, many identical copies, amplifying signal over noise. Then base incorporation emits a color captured per cycle. The number of cycles defines read length. This physical optical design is why Illumina produces billions of short, high fidelity reads per run. Now, primary analysis products and immediate QC you must do. So outputs are chromatograms, fast cube basis plus Fred quality. You then demultiplex, remove adapters and quality filter low confidence spaces or reads. Only then do you hand off to taxonomic or functional pipelines. Skipping these fundamentals guarantees false patterns later. Now, you use PacBio SMRT, which is different philosophy than cluster-based averaging. PacBio sequences single molecules in real time, recording polymerase incorporations as a movie, no cluster amplification. This preserves long context, but initially came with higher raw error, later mitigated by consensus methods. The conceptual win is contiguity. Then ZMW is how PacBio captures single molecule events at scale. The instrument uses Zero Mode Waveguides, ZMWs, nano chambers where a single polymerase, that is DNA complex, emits fluorescence upon nucleotide incorporation. Thousands of ZMWs run in parallel, each tracking one template. This architecture enables thousands of long reads concurrently. PacBio, what you gain or lose. So pros of using it is that it reads in the kilobytes to tens of kilobytes range, better assembly contiguity, improved resolution of repeats and structural variation, suits metagenome assembled genome, that is MAG, recovery and strain analysis. Cons of PacBio are historically higher per read error and cost. You often rely on consensus or hybrid strategies to maximize the